at this year's ASH 2019, we will be presenting a phase 1b study with a novel antibody-drug conjugate that targets CD123. This is an agent called IMGN632. The toxin payload used in this agent is of particular interest. This is a alkylating toxin, and it comes from a family of alkylating payloads called DGN that induce single-stranded breaks, as opposed to many of the known toxins such as PBD that induce double-stranded breaks. And we think the benefit of a single-stranded break induction is that we will see less neutropenia and better count recovery. And that was the reason that this next-generation toxin was selected, and we started these trials. It's a multi-center trial across the United States with a few sites in Europe as well open, and we're looking at 2 study populations in this phase 1b that I'll present. These are relapsed refractory AML, any age and any relapsed refractory, as well as patients who have BPDCN, which is blastic plasmacytoid neoplasm, expresses very, very high CD123 and a good disease area for evaluation of such a drug. In general, what we see, we looked at 6 different dose levels with a Q3-week dosing of the IMGN632. This is an antibody-drug conjugate, so unlike many of the bispecifics, it actually can be given outpatient. The infusion is about 60 to 90 minutes. It's repeated every 3 weeks, so a very convenient schedule. We don't see any CRS, cytokine release syndrome. We don't see significant edema, problems with albumin, creatinine. So I think logistically, this is a drug we think can move into clinic in the future. Tolerability-wise, the main thing we saw is that there were 3 episodes of what we call VOD among 95 treated patients. So that's about a 3% incidence. All of the VODs were reversible, none led to fatality, and they were moderate grade based on the grading system. And all of them were seen at the dose levels 4, 5, and 6. So we took this data and reviewed it carefully, and we've decided that we will be focusing on dose levels 1, 2, and 3. What was interesting is we saw responses at all dose levels, so we're comfortable using the lower doses where there was no VOD seen, which were also effective. And the overall response rate with this combination is about 25% CR/CRi in the relapsed refractory AML, which is quite decent. Of course, this has to go into combination like most things in AML. And what was quite interesting is in patients who had relapse phenotype rather than those who were refractory, the response rate was higher, about 35–40%. So now this study is going to be moving forward into combinations. This is a study I'm quite excited about. It will evaluate 3 different combinations: IMGN with venetoclax in relapse AML, IMGN with azacitidine in relapse AML, and then the one that could be really the most important, which is azacitidine, venetoclax, IMGN in older patients not fit for induction. Of course, we will be using a very low dose of IMGN, and we may need to adjust the venetoclax duration for the myelosuppression. Also, we're looking at MRD targeting with this drug. CD123 is a very important stem cell marker in leukemia, and for many years there has been interest in targeting CD123, and we think that this toxin payload and this drug has the potency to potentially eradicate MRD as well as leukemia stem cells. So one of the cohorts in this new study will be looking at MRD positive patients after planned induction consolidation, either with chemo or with HMA venetoclax, and see if we can eradicate MRD. And importantly, when we eradicate MRD, does that result in a much better event-free and overall survival? So we will have more data in the next 1 or 2 years with these combinations with the IMGN632.